Oh boy, it is like Christmas for me when we get to this point after a year of biochemistry. This last learning unit is integrative cellular biochemistry, where we really talk about, you could even call it physiological biochemistry. Now, I'll highlight that a lot of what we're going to talk about in this learning unit is not really new. You can sort of think of it like a meta analysis. We're taking a lot of the pieces that we've learned, maybe in a very detailed way in individual learning units, and we're really taking that closing 30,000-foot view and putting a lot of these pieces together. So the videos might be a little bit on the longer side because I really am doing this meta-analysis and putting things all in one place. Only about 4 to 5 videos that we'll have in this learning unit. But again, that's why the final exam really focuses on this. It's that high level 30,000-foot view of integrative cellular or integrative metabolism, integrative biochemistry, and thinking about how all of this comes together. So, like I said, it's like Christmas. Are you ready? Here we go. All right, so the first slide here is just going to sort of remind you of how we've studied biochemistry. We've looked at both eukaryotic and prokaryotic sort of systems, and we'll talk about what we mean there. And we think about, cellular biochemistry occurring in both unicellular and multicellular organisms. So I'm just going to tie this together and remind you of some things that I want you to think about. Typically, when we think about unicellular organisms, we typically tie that together with prokaryotic systems. And I want you to think about sort of the prototypical sort of prokaryotic organism that we've talked about, E. coli, right? We use E. coli in lab. Unicellular organisms are very easy to work with. You can grow them very quickly, all kinds of things like that. So typically we think about unicellular, we think about prokaryotes. Multicellular, we typically think of that sort of applying to eukaryotes. And we talked about this at the very, very beginning of biochemistry, but I want you to think about sort of the typical multicellular eukaryotic systems that we've talked about, right? We talk about ones like C. elegans, so those are worms. We talk about Drosophila, those are flies. Then you move into things like fish, and then you move into mammals, and eventually getting to humans. And so we can sort of think of that hierarchy in terms of becoming not only more like us. I mean, we're ultimately interested in understanding how biochemistry relates to disease in humans. But when we think about what we can study in the lab, right, we want it to be most like us, but clearly we don't want to be doing lots of experimentation on humans. So trying to find sort of that middle ground from an experimental standpoint, writing down sort of what we've said. We talked about thinking about worms and then flies, and then thinking about sort of other invertebrates, and then thinking about some vertebrates. And so those vertebrates might start with things like fish, and then eventually they can get to mammals, and then eventually to primates or even humans. So the last little piece to tie together here is it's very black and white to think that unicellular must mean a prokaryote, and multicellular is only eukaryotes, although that tends to be the case with multicellular. But I want to just sort of tie together an important piece that a lot of people will see studying, is that when you have a unicellular eukaryote, yeast represents sort of this perfect hybrid, if you will, of an organism that's easy to work with because it's unicellular, you can grow, and all of that, but it's eukaryotic. So it has those organelles and all kinds of things that are more sort of human-like in terms of studying cellular biochemistry. Plus, your lab's going to smell a lot better if you're growing yeast rather than E. coli. But at its core, what we know is that the basic biochemistry is the same. So when we think about similarities that we have with all of the cellular biochemistry that you've learned, that basic biochemistry is the same. So that basic biochemistry is the same, right? And that's why we sort of learned this in a very hierarchical sort of sequential way. Sort of we learned about glycolysis first, then we learned about pyruvate dehydrogenase, the citric acid cycle, electron transport, oxidative phosphorylation, right? That was the first half of our Biochem 2 course, is learning about those. That cellular biochemistry is fundamentally the same between multicellular and unicellular organisms, prokaryotes and eukaryotes, but sort of one of the main differences that we talked about is when you need to generate gradients and you are a prokaryote, you don't have organelles to do that. So you use your cellular membrane in order to do so. So again, the main difference, and when we do these 30,000 foot views, we're often going to oversimplify things to be able to sort of have them in this black and white or dualistic sort of way. But the main difference that we've talked about is the ability to do that compartmentalization. And when you have that compartmentalization, that is going to be, again, a main difference between the prokaryotes and the eukaryotes. And that allows us then to think about how we can not only compartmentalize for regulation, but compartmentalize or have a lipid bilayer, if you will, that you can sort of generate those gradients. So again, we've got that compartmentalization. That's going to be a differentiation when we have those organelles. And when we have multicellular organisms, we have the ability to do tissue specialization. And that's going to become an important piece that we'll have a separate video to sort of address. So we're going to think about tissue specialization. Okay, so I'm going to highlight here sort of the differences, and I'm going to bin these into these two categories. I'm not going to consider yeast. I'm going to consider this concept of unicellular prokaryotes. I cannot spell this morning. Unicellular prokaryotes. And then multicellular eukaryotes. And I'm going to highlight that our focus is really going to be, after this initial introduction, on multicellular eukaryotes, because we want to focus sort of on this tissue specialization piece. We've done this a little bit already, sort of talking about differences in cellular biochemistry between the liver and muscle, or between adipose tissue and liver, as we've learned about sort of different metabolic pathways. But we're really going to pull it all together here, and we're going to talk about different organ systems. You can sort of think of it as like being like physiological biochemistry. So let's just do a little summary here to talk about sort of some of the main differences that we think of here. I'm going to talk first just about those unicellular prokaryotes, because that's not going to be our focus. It'll be a little bit simpler to think about. When we think about unicellular prokaryotes and how they do their cellular biochemistry, we really have to think about how they're able to build up gradients and sort of control flux of metabolites. So we consider gradients, and particularly we're going to be talking about proton gradients, right? We know those are important for the synthesis of ATP. And we also have sort of metabolite regulation. So we're going to have metabolite sort of regulation, where things are. And they have no organelles, so they're not able to use that lipid bilayer. So no organelles. So they basically separate things. That the only really bilayer that they have is the inside and the outside of their cells. So the inside and the outside of their cells. Okay, so one other piece that I'm going to sort of just introduce here as a concept that we need to talk about, and it'll become more obvious and relevant as we get to sort of this multicellular eukaryote piece, is we have to think about communication. So communication mechanism. And you might say, well, what do you mean communication mechanism with a unicellular prokaryote? One thing that we know happens a lot with bacteria is their ability to communicate with each other, almost like they were a multicellular system, right? And not going to get into a lot of details here, but we know that this process happens through something called quorum sensing. And basically, it's the ability of these unicellular organisms to put information molecules out into the environment that travel through the environment to reach other unicellular organisms and sort of communicate what's going on, and then allow all of those individual cells to function almost in a group think mentality. So really cool system. Not going to spend too much more time talking about it. Again, I'm going to go over to this multicellular eukaryotic side, because this is really where we're interested in sort of living. So I'm going to talk about these two pieces. How do they perform their cellular biochemistry when they need to establish and utilize gradients, and then they can control and regulate flux in biochemical pathways by controlling metabolite location. And so when we think about sort of the gradients piece, and then we think about metabolite location, we can control all of that because we've got organelles. So again, we've got organelles. And even though there's tons of organelles that we can focus on and think about for doing cellular biochemistry, two main ones that we focused on, at least in terms of thinking about cellular metabolism, right, is we talked about mitochondria. And when we had that learning unit, anything that was mitochondria, we talked about in blue. And then they have their cellular counterpart in plants. We've got chloroplasts. Right? And so the theory with both of those is those are ancient unicellular prokaryotic bacteria that had magic properties that then became engulfed by another prokaryote that provided protection, that endosymbiont theory. So those are really two important organelles that we've talked about. I'm going to highlight two other ones here. Let's choose maybe orange color here. Just other organelles that we've talked about. We've talked about the lysosome, and then we've talked about the endosome. And when we think about those, the things that I want you to think about is when we think about the pH of these, let me sort of separate these a little bit here. When we think about the lysosome is going to have a high pH, and the endosome is going to have a low pH. So able to sort of control the cellular biochemistry by putting sort of things within these sort of different pH environments. So compartmentalization provides a really important piece to doing cellular biochemistry in eukaryotes. And again, we've got the unicellular eukaryote and yeast that are able to sort of monopolize on this. The last big piece that I want to talk about, and then I want to review a little bit of something that we've talked about before, is this communication piece. So when we think about sort of communication in multicellular organisms, we want to think about a couple of different pieces. We want to think about how we can communicate with hormones, and we would call this the endocrine function. So we're going to talk about the endocrine function, right? That's going to be communication with hormones that are put out into the bloodstream. So again, this is going to be hormones. We want to think about sort of the nervous system. Right? And we're going to use neurotransmitters and nerve impulses, which also rely on cellular gradients. So the nervous system is going to have neurotransmitters. And it's also going to have gradients that it utilizes for nerve impulse transmission, okay? And highlighting that for particularly this hormones piece, right? Let's highlight this maybe in blue here. We're talking about that endocrine function. We need to be thinking about the bloodstream as a way to sort of circulate those hormones. So the bloodstream, we need to have a circulatory system in order to sort of, communicate all of this information. And then the last piece that's going to tie into the very last sort of piece here I have here on a new slide to sort of do a summary is we are going to do a review of the HPX axes that we've talked about, right? That endocrine function and those hormones that are important. Again, this point in the semester, we're not going to review and understand all of the hormones that we learned about when we had that endocrine function sort of piece back in Biochem 1. I'm going to remind you of some threads that have carried through Biochem 2 that I do want you to know about, and some pieces that you're going to need to know. So as we sort of think about what we learned about for sort of the HP axes, here's what I need you to remember. I need you to remember that H stands for the hypothalamus. And I want you to remember that the hypothalamus is really responsible for sensing the external environment, and it communicates with the pituitary gland via nerve impulses. So this is sort of the piece that connects the external environment to the nerves to the nervous system. Okay, and that's going to communicate with the pituitary gland. So that's the P part of our HP axis. So the pituitary gland, right? We sort of learned about this as sort of the master sort of gland, the master hormone sort of regulator. The pituitary gland puts out lots of different hormones that go to sort of their secondary sites, and that is what that X refers to. So that X refers to where is that pituitary hormone going to? And even though these were all aspects that we learned about in our endocrine sort of learning unit in Biochem 1, don't need to know these right now. I'm going to highlight everything that you need to know come sort of final exam piece. I do want you to remember that, and here's the 3 axes that I want you to remember. We've got the HPA axis, which has to do with the adrenal glands, the HPT axis, which has to do with the thyroid, and the HPG axis, which has to do with gonads. Okay, and then here's the hormones that you need to remember and the disease states that I want you to think about. Now I'm going to highlight over here the HPS axis. I'm less concerned that you remember that axis specifically, but I do want you to remember the 2 hormones that are secreted by the pancreas that are really important to sort of cellular homeostasis here, particularly glucose homeostasis, and that's glucagon and insulin. Again, we're going to take a deep dive on those in just another minute on another sort of video here. But I do want you to remember that adrenaline and cortisol are the really important adrenal hormones that are important for the HPA axis. I want you to remember that the HPA axis kind of regulates your stress response, your fight or flight response. And here's the 2 disease states that are connected to that. With 2 situations here, the HPA axis and the HPT axis, I want you to think about what hyper and hypo sort of levels of these hormones have to do with. So we're going to be talking about adrenaline and cortisol. So high levels leads to Cushing's disease. Low levels leads to Addison's. And then the HPT axis, I want you to think about this as being your metabolism arm, right? You are sending out signals to control your thyroid release of thyroid hormones, T3 and T4. I don't need you to remember those specifically for your final exam, but I do want you to remember that the HPT axis is important for regulating metabolism. And if you have a hyperactivated sort of HPT arm, that is Graves' disease, and then Hashimoto's leads to a sort of a lowered level of that HPT axis, right? And so you have hyperactivated metabolism, hypoactivated metabolism, okay? Hyperactivated sort of stress response. Again, we're going to see that as it relates to glucose homeostasis in a little bit, right? And then hypo. So again, remember hypo, and then I want you to know the names of these 4 diseases. Wow, I know that was a lot, but sort of, introducing us and taking us through this integrative metabolism last learning unit in biochemistry.